Hello, everyone. This is our very first seminar in our brand new Western Biology Evolution Seminar Series hosted by me, Matheus Sanita. I am a PhD student here at Western University, and today I have the pleasure to speak with Dr. Rebecca Batstone. Dr. Rebecca is based in McMaster University. She did her PhD at the University of Toronto and her master's at Memorial University in Newfoundland. So the Batstone Lab um, combines experimental evolution with various genomic techniques to study how mutualistic microbes adapt to hosts, to understand how changing abiotic conditions modulate such adaptation, and to identify the genomic mechanisms underlying symbiosis evolution. So, Dr. Rebecca, I must say I'm very excited to hear all about your research, and I would let you take it away.
Awesome. Yeah, thanks so much for the invite for this. It seems like a really important effort in terms of, having sort of evolutionary biologists speak about their research and making it super accessible. So not just to others in your department, but anyone who has the link to YouTube can view this. So I think that's a really important effort. So thank you. Yeah, so I'm going to dive right in here. And I'm, introduction a little bit. So I am an assistant professor. I've been at McMaster University now for about a year and a bit now. And so, just getting things sort of running. But I do have probably the first few slides, I'm going to talk a little bit about what we've been doing in the lab currently. So I'm going through this phase where I'm trying to brand the lab and try to really think about the components or the sort of the framework in which we apply. So even though we work on a lot of different questions, like different biological questions, they all kind of fit into this framework. So what I'm calling the lab right now is the M3 symbiosis lab, or the M3 lab. So the M3 stand for multi-genomic, multi-scaled, and multi-mode. And I'm hoping, without giving too much of what I mean by those things, hopefully by the end of the talk, you're going to sort of hear these themes come up in the research that we do in the lab. So that's what we're going with. But just to kind of give a really broad scope of like the different approaches we apply in the lab. So Matheus already said we apply various sequencing techniques. So we do a lot of whole genome sequencing so we can resolve these beautiful multi-part type genomes in our microbes, which I'll talk a little bit about. We're also getting more into metagenomic sequencing, as well as some single cell targeted amplicon sequencing, hopefully actually in the coming years. We often will apply experimental evolution approaches to really dissect things like selective drivers and then quantitative genetics. So we also do a lot of genome-wide association studies where we're growing, a bunch of plants and different microbes, and we're able to use the genomic variation in microbes to predict phenotypic variation in plants. So those are, we do have other things that we do, but those are kind of the main approaches. And I did want to highlight, first of all, just because I've had such an awesome year working with students, and so I kind of wanted to give them a little shout out about previous research that we did last year. And so these are undergraduate students who worked with me, and this is, we went to the Ontario Biology Day where they're able to present their work. And so two of my students, Adham and Lachlan, were interested in understanding how interstrain competition, microbial competition, alters the legume rhizobium symbiosis. So they did a bit of experimental work and sort of some troubleshooting, so we can kind of get a system up and working in the lab. And actually it was great because these students contributed to a little write-up that we did in Current Biology. So this was a dispatch article. And so it was really great because they were already working on this project. And then I got assigned to do this. So we sort of collaborated on that. So that was super fun. My student Mathuja was really interested in understanding molecular dialogue. So the early signaling molecules exchange between the plant and rhizobia. And so that's actually we're trying to build that project more and more in the lab, and we are actively recruiting students to work on that project specifically. And then Sina was working on hydroponic growth, which obviously is sort of this, sort of hot topic in agriculture right now, and seeing how hydroponic growth might actually impact plants that heavily rely on symbionts. So a bit of a snapshot there. I do have graduate students in the lab. They're all working on various projects. So we were looking at biofilm formation and its impact on plant drought responses, antimicrobial resistance genes and soil microbiome communities, as well as we're starting to dabble into phage rhizobia interactions, which is a really exciting project, and seems like there's actually quite a few researchers in Ontario and at McMaster specifically that have a great expertise in phage. So we're trying to capitalize on collaborating with others who are more familiar working with phage. But okay, so that's kind of the snapshot of the projects. But I do also want to sort of highlight sort of one of the main mottos, or if you will, that we apply in the lab, is that celebrating the wins in science, no matter how small that they might seem, is kind of the thing that we do. So we always take time to really spend time together, celebrate, hang out. For example, we went to a battle axe throwing bar over the summer, and I found out quickly that I am terrible at throwing axes, but my students were amazing somehow. I don't know what skill set that's really requiring, but I didn't have it. And so, having fun with one another, and also my students have already contributed to lab success, so award-winning talks, and all of these sorts of things. So that's really important to highlight. And then in terms of future opportunities in the lab, so I am going to be posting an official advertisement for this probably in the next few months, but it's looking for a postdoctoral researcher to work as part of this Genome Canada grant. And so this is, I have pictures of the two leads here, Dr. George DiCenzo and Ivan Oresnik, but they are representative of like a team of, I think there's like 13 PIs on this project. So McMaster, University of Ottawa, University of Manitoba, Alberta, Queens. So we're kind of like, across Canada. And this particular project is all about how can we reduce the amount of fertilizer that we're adding to the environment, and as such reduce greenhouse gas emissions using microbial inoculants. And so can we get culture, locally sourced microbial inoculants that we can then use to improve crop yield without the use of all these fertilizers? So if you're interested in that, just keep this on your radar, and I should probably post the advertisement on Twitter at some point. And then I'm also looking for a master's or a PhD student to work in projects related to molecular dialogues as well as experimental evolution. Okay, enough of the plugs, but so I'll get into the actual science. Okay, so if you've ever heard me speak, I often start with this picture of that Darwin painted for us in Origin of Species. And so I'm going to read the quote. So it is interesting to contemplate a tangled bank clothed with many plants of many kinds, with birds singing on the bushes, various insects flitting about, and with worms crawling through the damp earth, and to reflect that these elaborately constructed forms, so different from one another and dependent upon each other in so complex a manner, have all been produced by laws acting around us. Okay, big long quote, but what it really paints is this picture of these multicellular eukaryotic organisms, which I refer to as macrobial organisms. And we're all very familiar with this picture as evolutionary biologists, but for those of us who study the tinier organisms in the world, microbes, if these microbes were visible to the naked eye, then we might imagine that this description would look something more like this, which I'm going to refer to as Leeuwenhoek's secret garden. And so it's the idea that microbes are everywhere, and they're often microbial symbionts. So they're intimately associating with these microbial hosts in ways that actually impact their evolution, so impact fitness. And so, we would add to that description these microbial symbionts living on and within these elaborately constructed forms. And more importantly, the fact that we haven't fully integrated as sort of like a, evolutionary biologist and the field of evolution, we haven't fully integrated how microbial evolution kind of fits into the paradigm of evolution that we have. So given this, general understanding of how macrobial organisms evolve, we need to integrate how microbial organisms evolve in order to understand fully evolution acting in the natural world. And that's my pitch for you today. And so I'm going to highlight a couple of features of microbial evolution that can actually differ quite a bit from what we sort of model or what we think of in terms of evolution in the macrobial world. So in microbial evolution, there's often an important component of horizontal transmission. So for example, if you're a host, you can environmentally acquire your microbes within the same generation. So you can acquire microbes that actually impact your own function within that generation. Zooming in a little bit more, microbes themselves often evolve by picking up mobile genetic elements that contain genes that can encode completely different functions. So this is known as horizontal gene transfer. So a microbe is picking up DNA from non-vertically being inherited from either the environment or from other microbes. Okay, so that's the first component, is we have this horizontal transmission. Another sort of aspect about once we take into account the microbial world is that a lot of the traits that we care about, so just to name a few, the breakdown and uptake of nutrients such as lactose, plant growth or crop growth, which is obviously what our lab studies a lot, pathogen resistance. So these traits are not just the trait of the organism in which we're studying, right? So it's not just the trait of a plant, because they're at least in part influenced by microbial genetic variation. And so because of that, they can be considered symbiotic extended phenotypes. So they're being expressed by the host, but they're partially governed by genetic variation in the symbiont genomes. Okay, so this means that symbiotic microbes can be important, although often unaccounted for sources of intraspecific variation in microbial traits. Okay, and then the final component I want to talk about is this idea of nestedness or multi-scaled symbiosis. So this is that it's often the case that mobile genetic elements such as plasmids or viruses tend to be nested within microbial cells, which then in turn are nested within macrobes. So the symbiotic extended phenotypes are emergent properties of these nested interactions. So you have loci within the mobile elements that influence not only microbial traits, but also macrobial traits. And those could be really important ones, such as those involved in host immunity, for example. So the pitch here is that if we want to understand and predict how the traits evolve that we all really care about, the charismatic traits that we measure at the macrobial level, we need to explicitly consider the multiple interacting genomes that give rise to those traits. And how do we do that? It's very complicated, but what you need is a nice steady system to dissect those features. So I've been focused on the symbiosis between leguminous plants and nitrogen-fixing bacteria known as rhizobia. And it served as a powerful model for understanding the ecology and evolution of host microbe interactions, but I would also argue for nested symbiotic interactions. So if you're not already super familiar with this system, leguminous plants such as beans, peas, alfalfa, they are high in nitrogen, not because they just are great at having nitrogen in their tissues, it's actually because of their symbiotic interaction with rhizobia. So the legumes actually house rhizobia within nodules located on their roots. And those actually form, every generation, and they develop over time on the plant. And in return, the rhizobia are able to convert atmospheric nitrogen into a bioavailable form, often as ammonia. And that's directly exchanged with the plant for plant-based photosynthate. So there's this beneficial exchange of resources, meaning that this symbiosis is often characterized as a mutualism. But one thing that is, to me, fascinating about the rhizobia end of things is that they often have these multi-partite genomes. So they have a large chromosome, and so if you study microbes, this large would actually be relevant to you, but it's about 5 megabases. But then they'll often have one or more symbiosis or plasmids that can actually be quite large. So in some cases, they could be like 1.3 megabases, 1.5 megabases. In most cases, it's the genes that underlie symbiosis, so the genes in which we have done knockout studies, so we delete the gene, that actually impact the ability for the strain to infect the plant and to fix nitrogen. Most of those genes are actually found not on the chromosome, but on mobile genetic elements such as plasmids. In other species, it could be found in integrative and conjugative elements. So that's sort of an interesting question in and of itself, is why are these genes essential for symbiosis found on these mobile elements rather than the chromosome? But it is a feature that I'm going to return to quite a bit throughout my talk. And of course, these genomes should be thought as dynamic. So in some cases, we find the symbiosis genes on the largest plasmids within the cell, sometimes smaller ones. So there's a lot of sort of dynamism and differences among strains that you're looking at. Okay, but if we think back to sort of what underlies this interaction, it's really, you could simplify it as the exchange of carbon for nitrogen. And so then you can imagine that if we're going to change the abiotic environment, such as by adding nutrients, we know this not just for the system that I'm working on, but for many other systems, such as in corals, nutrient loading can have a great impact on symbiotic outcomes. And in some cases, it can completely dissolve a mutualism. So think of coral bleaching, for example. And so this is well known, and it's not just specific to legume rhizobia symbiosis. But when we think of the abiotic conditions that are quite relevant to this system, well, let's think of agriculture, right? So oftentimes we're adding vast quantities of external fertilizer to our soil in order to increase crop yield. And so you might imagine that for plants that are heavily reliant on their microbial symbionts for nitrogen, that could impact the symbiosis, right? So you can predict maybe that it could disrupt the outcomes as well as the coevolutionary trajectories of symbiosis. And that is what the question that my postdoc advisor, Katie Heath, and her collaborator, Jen Lau, were really interested in understanding. What is the long-term evolutionary impact of adding nitrogen in a system on the legume rhizobium symbiosis? So I'm going to go into a bit of detail about the background of this project that I came in as a postdoc and have continued to work on since. So Katie and Jen and their research teams capitalized on this beautiful setup that they have at the Kellogg Biological Station in Michigan. If you haven't already been there, lots of really well-known projects that go on at the long-term ecological research site. But the one that's most relevant is that at this site, they have these experimental plots, and they're paired designs, or they're split plot design. So they have plots that have been fertilized with nitrogen going back to 1988, And why that's a significant date, it's the same time that Richard Lenski set up his experimental evolution study. So, 35-ish years now, a little over 35 years. But the paired plots right next to those ones that have been receiving nitrogen have been left without any additional fertilizer. So these are the ambient or control plots right next to it. So Katie and Jen went out and they collected soil from multiple replicates of each of those plots. And then they brought that soil back to a greenhouse and grew up a bunch of plants. So this is trifolium, clover, and basically you're using the plant here to do all the heavy lifting for you. If you're a microbial ecologist or microbiologist, it's often really hard to take or get one single strain of interest from a soil microbiome that probably has thousands of different strains. Plants are really great in this case because they have this symbiotic relationship with rhizobia. They're often used for trapping, so to actually isolate rhizobia from soil. And so that's what they're using these plants for, is to trap those rhizobia and then to isolate rhizobia from the nodule. So they ended up with about 35 isolates from the control plots and 28 isolates from the nitrogen addition plots. And so they took those isolates and then they conducted a single inoculation study. So this is where they have a new crop of plants, okay? And then they give each plant a single strain. So a strain from the control or from the nitrogen added plot. And then without giving the plant any other source of nitrogen, they're able to test, well, how well does this particular plant do with this strain? So how large does it grow? How much nitrogen content is in the leaves? Are the leaves nice and green? So you can look at these sort of proxies for what we refer to as partner benefit or partner quality. And what they found quite robustly is that when plants had been inoculated with strains from the control plots, they looked like what you'd expect, where the plants are growing really, large and lots of green leaves. Whereas the plants that were given strains from those nitrogen addition plots grew much smaller. Their leaf tissues didn't contain as much nitrogen. The leaves were yellow in color. So this sort of suggested that there is this effect of long-term fertilization whereby it's actually decreasing the benefits the plants are receiving from the rhizobia. But when they dug into this further, they did whole genome sequencing on all of those isolates, both from the control and the nitrogen supplemented plots. And they did a few different analyses. I'm just going to briefly talk about one here, where they're basically looking at the FST outlier analysis. And what they found is signatures of positive selection acting on genes within the symbiosis plasmid that were indicative of the shift to being lower quality due to being exposed to nitrogen seem to be adaptive. So there's this sort of adaptive signature to being low quality when you were a rhizobia from one of these nitrogen added plots. Okay, I'm very much simplifying things here, but I would be happy to answer more questions about this particular analysis. Okay, but one thing that they couldn't get at, because they didn't have the original strains going back to 1988 when they first set up the plots, it wasn't completely clear what the selective agents were that were responsible or driving that shift in partner quality in the rhizobia. So we could think of a few non-mutually exclusive hypotheses, okay? So first of all, it could be just this direct effect of nitrogen on soil. And we do know, in particular, some sources of nitrogen can actually change the soil pH, often by decreasing it. Can have all these cascading effects on the soil chemistry. And that in turn can have these direct effects on rhizobia within the soil, because they do live, they have a dual lifestyle, right? They're living in the soil as saprophytes, and then they spend time with the plant in symbiosis. So it's during this sort of free living phase where they might be directly impacted by the addition of nitrogen. And for some reason, this happens to increase the frequency of these lower quality strains. Okay. Another hypothesis would be indirect effects of nitrogen. So this could be through the plant. So if the plant now has a cheaper source of nitrogen in the soil, supplied through the fertilizer, then it might not invest into symbiosis as much. And we do have good evidence that on a very short term, you provide nitrogen to a plant, it's going to form fewer nodules, because nodules are really expensive for the plant. So if it could get nitrogen elsewhere, it won't form as many of those associations. And so you can maybe expect from that, then the plant is then relaxing selection for these higher quality strains, and that can allow the increase in frequency of the low quality strains. So those are, again, the ones that are being selected. Okay, so how do we go about teasing these hypotheses apart? Well, enter me as a postdoc. When I came in, I had already done or applied an experimental evolution approach during my PhD. And so what I wanted to do is take my set of expertise and use this framework or this approach to dissect those selective drivers. And so this is what I did. So I'm going to walk you through the setup of the experimental evolution study. So I started with 3 populations that I made from those original isolates that I was talking about that were isolated from the field soil. Okay, so I had one population that was all strains coming from those control plots. So it was 28 strains from the control plots. Another population that was all strains from the nitrogen supplemented plots. And then I had one population that was a mix, an equal mix of both. So 14 from control, 14 from unsupplemented. Okay, so 3 different populations. And then I exposed them to either high nitrogen, trying to also reflect actually the amounts that had been added in the field. So I did make sure to add relevant amounts. Low nitrogen, I actually didn't apply any nitrogen, so that really is just an N-free treatment. And so I did that for both, for all of the populations. But then specifically for the mixed population, I added an additional treatment, and this was a plant absent treatment or soil only treatment. And so this allowed me then to dissect the direct effects of nitrogen versus the indirect effect of the plant, because now we're manipulating whether the plant's present or absent. I passaged these populations in a growth chamber for just under 1 year. And so what this looks like is you allow you inoculate all plants at the beginning with your respective populations. And we have, of course, multiple replicates. So the numbers at the end here is how many independent replicates we've included for each of these passages. And so the plants grow, they form nodules, and then we cut the shoots or the top parts of the plants off, and that induces nodule senescence. So the nodules break open, rhizobia are released into the soil. We have a fallow period, kind of like what you'd see in sort of like a normal seasonal cycle in the field. So that's just, you know, the rhizobia are hanging out in the soil, and then we replant from the same seed stock again. So there's no plant evolution going on here. There's potential microbial evolution. And then we did that for 4 passages or growth cycles in total. At the very end of the final growth cycle, we created soil slurry. So this is literally just adding sterile saline solution to your soil for each of your pots. And then you're filtering any sort of particulate, so any soil particles. You're also diluting to get rid of any sort of chemical differences. Obviously, we have nitrogen in one versus the other, so we're diluting that away. We're basically just trying to isolate whatever rhizobia are present in the pots. And then we take that as a new inoculum that we use in a soil slurry experiment. So now we take the inoculum and we dispense it onto multiple plant replicates, and then we don't provide any source of nitrogen. We just see how well those plants grow and how many nodules they form. So that's the soil slurry experiment. And we measured various things. Above ground shoot biomass, it's often used as a proxy for partner quality. So like how well, in this case, it would be the population quality, right? Like how well does the rhizobia in this population do for the plant under limited N, again, because we're not supplying the plants with nitrogen. Nodule number and size, so how many associations did the plant form? How large were those nodules? The larger, the more indicative of a productive nodule. That's often the case. So they'll be fixing nitrogen. And so these could be thought of as proxies for host investment. And then rhizobia were isolated from nodules. So we dissected out 10 nodules per plant. So, given replication here, that means 2 to 4 plants per soil slurry, 3 to 4 soil slurries per experimental evolution treatments. We had multiple treatments. It ended up being thousands of nodules. And I wanted to put up Carla and Labake here because they were fundamental for actually doing this work, because it is very time consuming. Okay, so let's get into the questions that we set to address in this experiment. So first of all, can we even recapitulate the results that we saw from evolution in the field in our 1-year experimental evolution study? So to show you the results, I'm going to orient you first to, we have our 3 initial populations as each of these panels. So our control, the mixed, and the nitrogen added. On the y-axis, we have a proxy for host benefit. This is shoot biomass. And then along the x-axis is the legacy effect, or the experimental treatment effect when we're doing the experimental evolution of nitrogen, so presence or absence. So it's not what we're currently manipulating in this experiment, it's the past effect of nitrogen on the populations. And so adding some data here, so every point is representative of a independently passage replicate. So that's 2 to 4, a mean of 2 to 4 plants for each of our soil slurries. Okay, so now filling in all of the data here. So basically what we found is there was no significant difference overall in our control population and our nitrogen addition population. So those are strains that just came from one of the 2 plot types. But in our mixed population, we actually did recapitulate what we saw in the field. And so just to remind you what the results from the field study were, is that they have plants inoculated with the strains from the nitrogen added plots grew worse than those inoculated with the control strains. And that's pretty much what we found in our mixed population. Okay, so for one of our 3 populations, we were able to recapitulate what was seen in the field, and rhizobia populations exposed to high nitrogen seemed to be less beneficial. Okay. But if so, do we see the same response when plants had been absent during experimental evolution? So this would mean that if responses are the same, regardless of the plant treatment, then they're likely due to the direct effects of nitrogen addition on rhizobia. So showing you the data here, so this is just for the mixed population only, and then the panels represent whether the plants had been present or absent during experimental evolution. And so the take home here is that we basically don't find any difference. When plants had been absent, the populations are just as good or bad on the plants when they were exposed to nitrogen or not. However, when plants had been present, we actually then find the signature that was found previously in the field. So plants inoculated with those strains exposed to nitrogen are doing worse. Okay, so this seems to us that hosts rather than the direct effects of nitrogen are the selective drivers for this mutualism decline. But what that doesn't get at, this is sort of the, the broad sort of, organismal level signature of this. But what about the underlying mechanisms driving it? And we can think of, again, some non-mutually exclusive factors here. So first of all, there could be simply just overall differences in the abundance of rhizobia present in each of our conditions. So we can imagine that under nitrogen addition, or when hosts had been absent, that plants are going to form fewer nodules, and therefore fewer rhizobia are going to be surviving from one generation to the next. So then there's just fewer rhizobia at the end. We inoculate that onto plants that aren't given any nitrogen, and they're not going to grow as well. Okay, so we can actually get at this question using the results from our soil slurry experiment. And so this is very similar to the plot I showed you before, except for now on the y-axis is the number of nodules that the plant formed, or the number of associations with rhizobia. And overall, we're not seeing any big differences or any significant difference between nitrogen versus control or N-free manipulations in the soil. So it's not like rhizobia were less abundant when nitrogen was present. Okay, so what about the difference in the relative abundance of rhizobia? So we started with 28 strains initially. How did the composition of that population change over the course of our experiment? And we can get at this using whole genome sequencing, or at least that's the approach that we took for this. I'm going to go through this pretty quickly, but I want to highlight here that what was really great about this is we had these beautiful hi-fi long read sequences of all of our 56 ancestral strains. So these are the ones that we put into the experimental evolution study at the beginning. So they're the ones isolated from the field plots. But then we did a bunch of short read sequencing. So this was 363 derived isolates that we isolated at the very end of our experiment. And so we, first of all, we made sure that they were all rhizobia. So we did some 16S Sanger sequencing to make sure. And then we triple purified them, because you have to be really careful about not introducing any intra-isolate variation if you're doing short read sequencing. So we made sure we had these pure cultures, and then we used parent end sequencing with 100x coverage, probably more depth than we actually needed, but that's okay. So what this resulted in is about 39 to 47 isolates per experimental evolution treatment. And then from there, I had an amazing undergraduate student work with me in the summer, Sing Yuan. And so what he did is he completed de novo assembly for all of our 363 isolates using spades. And then he calculated the average nucleotide identity, or ANI, which compared each of our derived isolates to each of the original 56 ancestral strains. And he used the program FAST-ANI. This was completed on core genome. So we're not including genes that were present in some of our isolates, but not present in the ancestral strain, for example. And for most isolates, what turned out to be quite important for this analysis is that the top hit, so the original isolate, or the ancestral isolate with the highest average nucleotide identity to each of our derived isolates, was greater than 99.9%, whereas the second hit was less than 98%. So it means that our ability to say this derived isolate is most likely the derivative of this original ancestral strain, we have quite a bit of confidence in calling that. And our original strains were they're still all closely related. This is all still within the same species, but they were different enough that we could assign the most probable ancestor for each of our derived isolates. There was only one isolate where that wasn't the case, because it was likely just a contaminant, but that was the only one out of the 363 that we had to exclude. Okay, so going over some very broad results here. So based on the 362 derived isolates, we were able to assign them to 25 of the original 56 strains. So there definitely were, half of the strains survived throughout our entire experiment, but the other half did not. 17 of those strains came from the control plots, and only 8 from the nitrogen plots. So the strains that were originally isolated from those nitrogen plots did less well at surviving the experiment that we conducted. All I'm showing here in this plot, because there's a lot of detail, on the x-axis are those 25 most probable ancestors. And so one really cool thing is we can actually we could calculate like the relative fitness of each of those strains in our different conditions, and then see how consistent they are across our replicates. So there's a lot of information that we can get from that analysis, but I'm not putting this up to, walk you through it. It's just to say that, we can calculate these various parameters. Okay, but getting back to the actual question, so how did the relative abundance change in rhizobia? And in particular, we could ask the question of whether there might have been stronger selection for higher quality strains under, for example, nitrogen-free conditions and when hosts had been present. And the reason why we can get at this is because we have done single inoculation studies with each of those strains to see how well they do on each of our plants. So we can tell whether a strain is a high quality strain versus low quality. And so what I'm showing you here is on the y-axis, we have the frequency of high quality strains in our population. And the dotted line is the frequency that we started with in our experiment. So this was the baseline. So if we see a bunch of our data points above that line, it means that the frequency of those high quality strains is actually increasing throughout our experiment. And so you can see that in both our mixed population and our unadapted populations, there were more high quality strains present in our soil slurries than what we inoculated with to begin with, so at the very beginning. However, the opposite was true for the control population, interestingly enough. So we actually found less proportion of high quality strains in those populations. But when we zoom into that mixed population where we can actually dissect the impact of the plant, basically what we're finding is that there's this higher frequency of high quality strains regardless of host presence, which is actually a really interesting sort of point. And I'll elaborate on that in a second. So basically, we saw this increase of frequency of the high quality strains in our mixed population regardless of nitrogen or host treatments. And the reason why this is sort of an interesting result is because of sort of the paradigm that exists in the field of rhizobia and partner quality. We sort of expect that we have this underlying expectation that if you're a really high quality partner, a really great symbiont, you're going to be less great at competing in the soil. So in the free living stage, you're going to be a worse competitor. There should be some underlying genetic trade-off between the two. And so that's what I'm showing you here from this perspective piece that we wrote, or this based on a dispatch article. We're basically saying that there's this expectation for the trade-off, but when you actually go and look at the data that we've collected so far as a field, not just me, but like as a field we've collected, we don't actually see evidence for this trade-off. And that seems to be playing out in our results as well, where the high quality strains might not just be really great symbionts, they just are higher vigor strains overall, and they're better at competing. So that's why we're actually seeing an increase in the frequency of those strains, regardless of whether hosts were present or how much nitrogen was present. Okay. Another thing, though, we could look at is the amount of strains that were that survived to the end of the experiment. So strain richness, it's based on the strain count at the end. And we can compare that under nitrogen-free conditions versus host-present treatments. And so when we did that, first of all, across the board, again, in those nitrogen supplemented populations, we recovered very, very few strains, regardless of nitrogen. So that was one point, is that the richness was significantly lower from that population than the other two. But then when we zoom into the mixed population, what was really interesting is that when plants had been present, they're really driving down the number of strains. So there's a higher frequency of high quality strains, but there are fewer numbers overall. Okay, so lower strain richness under nitrogen-free conditions when hosts had been present, but we didn't see the same difference when plants had been absent. Okay, so putting this all together and going back to the original data that I showed you with shoot biomass from the soil slurry experiment, what we're sort of thinking is happening is that there's higher partner quality or higher frequency of high quality partners under nitrogen free conditions due to both of these things acting together. First of all, there's a greater frequency of high quality, high vigor strains, and stronger host selection for fewer of those strains, potentially reducing interstrain competition. So the plants are actually able to benefit more from the fewer high quality strains that are present when nitrogen had not been added in the experiment. Okay, so I'm going to wrap up here. So what we're working on now is the underlying genetic mechanisms of mutualism decline. And so, for example, we can compare directly how well the derived isolates do for plants, so how much benefit they provide, directly compare that to their most probable ancestor. And we might find, for example, maybe they increase in quality or decrease in quality or that magnitude of difference depends on what experimental treatment that they were in. So that's something that we can get at using single inoculation experiments, and it's something we have all the data for. We've already conducted that experiment. Another thing we can look at is the presence-absence variation among the derived isolates. So this would be genes within the genomes, and we can compare those to the most probable ancestors. So we can get at gene gain or loss through horizontal gene transfer, for example. And then finally, we can look at the number of SNPs. For example, if SNPs occurred, but only in particular treatments and not others, that would be suggestive of maybe SNPs underlying adaptation to those conditions. And so all of that information also will come to light soon. We have a lot of data on our plate right now. So we'll be writing that up probably as a separate paper from all of the experimental stuff that I just showed you. But just to summarize, so the legume-rhizobium symbiosis is a powerful model for identifying selective drivers underlying mutualism decline. So that's why I tend to use this system, and I probably will for quite some time. Experimental evolution approaches allow us to really tease apart these selective drivers and really get at how, in this case, microbial genomes might change over the course of that experiment. So if you couple that with sequencing, you're able to get sort of the printout of evolution occurring in real time, which is quite fun to look at. And then in terms of the conclusion, from our experiment, we think that rather than the direct effects of nitrogen, hosts are indirectly driving mutualism decline by being less able to select for and reduce the number of high-quality strains under the nitrogen supplemented conditions. And overall, a better understanding of the mechanisms driving mutualism decline will help mitigate the reduction of benefits exchange in mutualism, which we're actually seeing quite a bit, not just in agriculture, but in, for example, with corals and with other types of symbioses in which nutrients are exchanged. All right, so just quick acknowledgements to, first of all, my co-authors highlighted in orange here, or the PIs on this project highlighted in orange, and then other folks that are involved in this project that were essential for getting this massive data set. And then to various sources of funding. And then I also just want to thank Matthews for inviting me and for giving me the opportunity to talk a little bit about this amazing project that I've been doing. All right, thank you so much. So yeah, I'll take any questions.
Wow, what a wealth what a wealth of information, I must say. I really enjoyed your talk, Rebecca. And
Thanks.
I think, when I developed this project, the seminar series, I really wanted to showcase that evolution, or the study of evolution, is a multifaceted endeavor, right? So, because usually in undergrad, we always study evolution mostly in a population genetics way, right? So studying allele frequencies change over time in populations. But here today, you're really showing us that, evolution, there's much more to evolution than just allele frequency change. So this was great. And I do have a few questions. So let's see how that goes. So
Okay.
my first question for you is, at one point in your talk, you mentioned the core genome of the rhizobia, right? And I'm very interested in pangenomes. I think pangenomes, perhaps after horizontal gene transfer, pangenomes have really revolutionized the way we see microbial ecology in evolution. And I wonder whether, so because you also mentioned, mobile genetic elements are important in the evolution of those symbioses. So I wonder whether in the future you would like to study pangenomes, or the accessory genome, not just the core genome, but the accessory genome as well.
Yeah, absolutely. So that is in the very last slide of the next steps, that's basically what we're doing is a classic pangenome analysis based on our strains. So we're going to be looking at presence-absence variation of genes that have been annotated using actually different annotation pipelines, but ones that we we have the most confidence in, basically. So we're going to be able to determine how much of, the for example, in our strains that came from the nitrogen addition plots, we might find genes present from the control background, and vice versa. So we might see instances in the control background where now we see genes from the strains from the nitrogen addition plot. So that's like one full analysis that we can do given that data. But then we can also expand it. So instead of just looking at our, 363 derived isolates that are all like highly closely related, we can actually look at the broader database and see kind of how that fits into the pangenome of, strains isolated from different conditions or from the field. We also have a full collection and a data set on 2018. So all of the strains that I was talking about that we used in our experimental evolution, those were all collected in 2008. But Katie and Jen went back in 2018 and re-isolated rhizobia from the plots. And so we also have all of the genomes from 2018. So we can do pangenome analyses, figuring out if the genes of interest really are found on these mobile elements, and then where do they end up in different backgrounds. So it's going to be a lot of data to sift through and to figure out the story that the data is telling us, but yeah, that's what we're planning to do.
And also at the beginning of your talk, you showed us that rhizobia, for most species, they would have their main chromosome, let's say, and then they have a series of plasmids. And I think there is one plasmid which is called symbiotic plasmid or symbiosis related plasmid. So what type of genes would be in those plasmids?
Yeah, so why they would be called the symbiosis plasmid is because they often contain the canonical symbiosis genes that have like various annotations. So it's like NOD, NIF, FIX, like these canonical genes that have been used in going back years now, where they would knock out one of those genes and find it would completely disrupt the ability of the strain to form nodules with the plant. So a lot of those genes, canonical symbiosis genes, are found on the symbiosis plasmid. But then there's also genes, essential for living in the free living environment, for example, that could be found on a different plasmid. And there's some cross sort of even signatures of recombination across plasmids as well. So the story is a little bit more complicated than kind of how I'm simplifying it, but that's basically what it comes down to, is if we find those canonical symbiosis genes on whatever plasmid, we tend to call it the symbiosis plasmid.
So I think one concept you are really bringing is this idea of nested evolution and nested symbiosis, right? In which you start from mobile genetic elements, from the symbiont all the way to the plant genome. Those mobile genetic elements, when they are horizontally transferred, are they restricted to the symbionts, to rhizobia only, or we see that transfer also happening to the host?
That's a great question, because I was only talking about plasmids today, but let's think about viruses. Like viruses can certainly just interact with the host in various ways. So there's nested symbiosis, and that's really what I'm talking about, these the specific plasmids within the rhizobium cell that then infect plants. But I think as a broader concept, it's more of a multi-scaled symbiosis. So you can have mobile elements directly interacting with the host, which then impact their interaction with the with the bacteria, for example. So it can go in all different directions. It doesn't have to just be mobile elements with the microbial symbiont with the host.
Mm-hmm. And one last question, speaking again about mobile genetic elements, because I've seen the term MGE being used to almost like an umbrella term for any type of genetic fragment that is swapped around, including transposons or transposable elements. But in your case, I think we're talking mostly about operons, right? That can be swapped around.
I think in the case of where I'm talking about the plasmids, yeah. So it would
Or
be, you'd
plasmids, or
often
entire
see,
plasmids, yeah.
yeah, yeah. But I mean, because something might not have, like, you could kind of make a definition of, well, what is a minimal mobile element? Like, doesn't it have to encode, first of all, like some sort of origin of replication, some ability to transfer, right? So shouldn't it have to include those features for us to call it a mobile element? And I would agree, yes, but then that could break down, right? So if recombination, you might have, for example, like a gene segment, an operon, that becomes mobile because now it's been infected by some, I don't know, virus that packages it. So it's delineating like what is a mobile element, and like what features are actually fundamental to that. I think for me at least, it becomes a little bit tricky to parse out. But I think more generally, we're talking about, if we look to, basically it comes down to linkage disequilibrium in my mind. Like if we see things co-transmitting together for a long period of time, and then things that are like in completely different backgrounds, not due to just vertical transmission, then you can think of those also as mobile elements.
Mm-hmm. Absolutely. Well, again, thank you very much. I think, the talk was amazing. Q&A question, very insightful as well. I'm very happy that we're starting the seminar series with your talk. This was great. So
So
I
I'm so
will,
glad you enjoyed it.
absolutely. I will stop the recording, but we can also chat a little bit afterwards, okay? So
Okay, cool.
so thanks again, Rebecca, and thanks everyone for watching us.
Yeah, thank you.